Okay, I guess it just started.
Yeah. Okay, so maybe we can start. So I've started the recording, and this recording will be shared, so please feel free to keep your camera off if you would not like to be recorded, although we encourage as many faces and voices in participating as possible. So welcome everyone. Thank you so much for joining us this afternoon. I hope everyone has had a stimulating day of workshops and discussions. I know I have. My name is Lily Aleksandrova, and it's my pleasure to moderate this workshop today titled Translational Research for Novel Pharmacotherapies. I'm a postdoc in the UBC Department of Psychiatry in the Center for Brain Health, studying novel treatments for conditions like substance use disorder in Dr. Phillips's lab. I'll be leading the discussion portion of this workshop and monitoring the question and answer and the chat functions. We're hoping to keep you engaged and generate some conversation, so please feel free to share your thoughts and questions, and we'll be happy to address them. And questions can be asked either directly by unmuting yourself on Zoom, or through the WOVA question and answer feature, or through the Zoom chat. And I'll do my best to monitor all the platforms. And to get us going, if you would like, you could share your name, your role, and the region you're joining from in the chat. And next, I'd like to introduce my supervisor and mentor, Dr. Tony Phillips, who will take over for the presentation parts of this workshop. Dr. Phillips is a professor in the UBC Department of Psychiatry in the Center for Brain Health. He served as the founding director of the UBC Institute for Mental Health and as scientific director of the Psychiatry Institute of Neurosciences, Mental Health and Addiction for almost a decade. His pioneering research at UBC led to the discovery that dopamine plays an essential role in the neural control of motivation and memory, with clinical implications for understanding biological correlates of substance use disorders. In recent years, his lab has focused its efforts on translational research and the development of novel options for the treatment of substance use disorders. And if Tony, you could change the slide for me. Just go over the agenda for today.
Yeah, thank you.
Yeah, in part one of the presentation, Tony will introduce the concept of reverse translation. Yeah, let me try again. Of reverse translation and highlight, oh, there we go. And highlight some major challenges in addressing critical aspects of opioid use disorder. And next, we'll have a brief 5 to 10 minute pause for an audience poll and some discussion. And Tony will then present data obtained from in the lab, evaluating a novel non-opioid pharmacotherapy for opioid use disorder and pain management, demonstrating a reverse translation strategy in practice. And we will end with a 20-minute discussion section addressing questions and other comments that might arise. And I wanted to emphasize that whatever your background, whether it's preclinical, clinical, or lived experience or anything else, we welcome and value all perspectives. So with that, I'll pass it on to Tony for part one of the presentation.
Thank you, Lily. And everyone that's here, I just want to acknowledge that Lily's more than just a moderator of the session. She and I brainstormed on how best to organize this material, and she helped with the slides. And so she's a co-author of this effort. Thank you, Lily. Again, competing interests. I do hold a patent on, it's not one of the molecules we'll be talking about today, but it's related. And also we've started a company called Resilience Biosciences, focused on the development of tetrahydroprotoberberines. So I'm happy to disclose that. What I want to begin with is, I'm a preclinical neuroscientist who's very interested in applying the knowledge that we have of brain and drugs and their action on the brain to issues of social importance, such as many aspects of mental ill health, and in particular addiction and substance misuse, as it's more properly called these days. And normally the way in which people in my game proceed is beginning with basic research, finding some interesting molecules, some new insight into brain function, and then seeing how we might develop that further so that it could become a potential, in our case, a drug. And then, of course, once you've done that and you've proven that it's safe, you take it into the clinical setting to see whether or not it's going to have efficacy. First of all, you show it's safe, and then you show that it has efficacy in treating some aspect of mental ill health or substance misuse. I want to introduce you to another concept, which I think is more accurate, and that's the concept of reverse translation. And we're sort of engaged in that in our research efforts at UBC and also in this presentation. We quite often take our lead from what we observe in the clinic and what, the trained and sophisticated clinical assessment of these disorders. They're all complex, they're multifaceted, and it's the perspective from the clinic that actually leads us back into trying to find out whether or not we can marry our basic research with a clinical need through a translational research program. This idea of reverse translation is really gaining traction, and I think it's a much more accurate way of describing the discovery of new molecules that'll hopefully have efficacy in treating conditions such as substance misuse. So this is the approach we're taking here. So the background, we're not talking about all of mental health. Obviously, in this conference, we're talking about opioid use disorders. And I would claim that there's still a very, very real need for novel medications to be developed that adequately address at least three important aspects of the opioid use disorder condition. One is and I'll talk more about it in a moment, the concept of withdrawal, where a person voluntarily decides to undergo detoxification so they're no longer dependent on the opioids that they've been using. That's one issue. Another related issue is the fact that many people have developed difficulties in dealing with opioid use because they were prescribed opioids for pain management that they desperately need. And sadly, when they are given too much or for too long, then a dependency can be developed. So there's a huge initiative underway now to try to find new analgesics that are non-opioid. And this work I'm going to talk about today has a relevance to that too. And really the holy grail in this whole enterprise of finding drugs that have true efficacy in helping with opioid use disorders is the capacity to prevent relapse. Once someone has been brave enough to attempt withdrawal and has done that successfully and stays abstinent for many months, sadly, there are a number of factors that can precipitate relapse. And so we need to be finding pharmaceuticals that might address relapse. Now, there's a diagram in the right-hand side of this slide, my slide here, that captures the complexity of opioid use disorder quite nicely. It's from a close friend of mine and colleague, George Koob, who's the head of the alcohol and alcohol addiction unit in NIH. And so in this circular diagram, what is it that gets the whole process of potentially becoming dependent on opioids underway? Well, most people agree that it's the attractiveness and the mind-altering effects of experiencing an opioid, which can be done repeatedly, and then it can get into a stage where there's excessive intake, binging, which leads to intoxication. And then we enter this period that I talked about a moment ago, which is the desire to withdraw and no longer become dependent. So they go through a detoxification phase. And then that leads to abstinence. Has to be recognized about the detoxification stage is how multifaceted and how many aversive experiences arise from entertaining withdrawal. And I have a slide that will explain that a little bit further. And the withdrawal symptoms themselves can even become conditioned to the environment in which you're experiencing withdrawal. So you can get something called conditioned withdrawal. And all of these withdrawal factors can lead to relapse, which I spoke about a moment ago. So we're trying to break this cycle using pharmaceuticals, hopefully. So this is the goal one then, is to really emphasize the fact that challenges related to successful voluntary detoxification from opioids can be seen as a key step in gaining further clinical support to overcome opioid use. This really is a barrier, I think, and many other people think, to the successful, more integrated treatment on the journey to become less dependent on opioids. The withdrawal condition itself appears quite quickly in a matter of hours after the last dose of an opioid. And then remember that in this instance, the individual has decided voluntarily to enter this phase of detoxification. And so after short-acting opioids, you get these signs appearing within 6 to 12 hours. In more long-lasting opioids, where there's still some opioid residual material on board, it can be as long as 30 hours. But anyway, following this initial period, then you start to see the symptoms that I've been alluding to, the withdrawal symptoms that occur approximately 3 days after you begin this journey. And they're listed here, and it's really many forms of painful experience, also psychological distress, and then drug craving also is prominent. And the other thing that may be less appreciated is that this isn't just over in a matter of days. It's a condition that can be protracted. And so that places extra challenges on us. So the other thing to remember here is, I keep emphasizing its nature as a barrier. Well, direct experience from people with lived experiences undergoing withdrawal, they certainly provide testament to the fact that this is a deterrent to seeking appropriate clinical assistance, because they want to have these symptoms relieved by a new injection of an opioid. And as I said a moment ago, because these adverse effects are recurrent, they can contribute to relapse as somebody seeks to escape these adverse experiences through the use of an opioid. So again, this is one of the take-home messages I want to leave you with. I believe that the effective management of withdrawal in those people who really genuinely choose to change their lifestyle and not be dependent on opioid use, that this, if we could manage this properly, right now it's a barrier, but I think with proper management, it could become a gateway to effective treatment of opioid use disorders. Now, there's another concept that I want to introduce you to. There's a growing appreciation of it, just to highlight the close relationship between opioid use, obviously, and pain perception. It's now well recognized that as you enter into a period of opioid withdrawal and you do formal tests of your analgesic reactivity, there's an exacerbation of pain. That's really quite a prominent feature of the withdrawal situation. And it's sometimes formally called nociceptive, so pain perception sensitization. In animal studies, when you can measure it really formally, you can find that after a 48-hour withdrawal period, and especially after a second withdrawal experience, the pain reactivity can be 100% more sensitive. So this is really quite an important factor. And then prior dependence on pain-relieving opioids is also itself associated with increased sensitivity to painful stimuli. So the opioids are actually priming the pain systems in the body when they're not present. There's a rebound effect. And this could be treated a major factor in dealing successfully with opioid withdrawal. And that's what it says here. And that negative state, of course, could maintain somebody in a drug-seeking pattern of behavior. And it also underscores the fact that if we could find drugs that were not opioids that are required for the legitimate management of pain, then I think a lot of this problem would start to become less prominent. So fortunately, there are options. I was listening, it was rapt attention to some of the talks this morning. And of course, practically everyone was as properly paying homage to the fact that opioid agonist therapy, So where you simply substitute a legal opioid for heroin or something, fentanyl or whatever, that this is the most utilized treatment option for opioid use disorder, simply substituting one opioid for another, one that has more benign pharmacokinetics, etc. And there's a place for this. It really has helped, especially from a harm reduction perspective. But there's also an effort that I'm part of. So this lists what those long-acting opioids are, and I think everyone's aware of those. It's this next slide that I wanted to bring to your attention. This is the first non-opioid option for dealing with withdrawal. It's called lofexidine, and it's an alpha-2 adrenergic agonist. It was actually study in the 60s, and just last year, the FDA finally approved its use in facilitating detoxification from opioid dependence. However, it has a profound hypotensive effect, which can limit its use. So it's a welcome addition to the pharmacopoeia, but it's not without its problems. And the feedback from individuals taking it, it's not a very pleasant experience when you have these hypotensive effects. So here's the point, Lily, where we were going to break for a moment, because as we were saying in our pre-workshop discussion, I'd really like to know whether what is the general opinion about striving to find effective medications that can manage this whole detoxification journey? Is this a desirable goal? And then we put it out as a poll. Do we have any answers?
Yeah, so we seem to have 100% response rate.
Mm-hmm.
And we can see, so for the first question, is successful management of opioid-induced detoxification a desirable goal? And we have 11 answers, of which 75% now say yes, and 25% say no.
Okay.
And
maybe, sorry, can we, that's good. I mean, different points of view. It might be nice if we had a moment or two to hear about the pros and cons. So I think that's part of your plan.
Mm-hmm. Yeah, that's the third question. So in other words, we worded it differently. Are medical options for voluntary non-opioid assisted withdrawal worth pursuing? And we have a similar breakdown, this time 90% for yes and 10% for no. And we have 10 answers in total. And then we have the short form question, which was, what are some potential pros and cons of non-opioid options for substance use and pain management, and what are some potential challenges? And we have 5 answers in total. One says potential concerns may be the decrease in tolerance and increased risk of overdose upon relapse.
Yeah. And that's a very legitimate point. Yeah.
Mm-hmm. Yeah.
Okay.
And then one says major pro, reducing suicidal ideation with ketamine. So we know more in the context as a novel antidepressant, but also in the context of addiction and substance use disorders nowadays, ketamine, and with this rapid action as well as efficacy in reducing suicidal ideation. And they say that ketamine and cannabis may reduce use for some patients. And another comment echoes the utility of cannabis as an option for some patients, and that the health impacts are less severe and it's more available. And then we have, as a pro, may see a decrease in use, but also in inadequate pain management. So again, bringing back to Tony's point of this dual action on substance use and pain. And then as a major con, we have access and racism, which are obviously barriers to effective treatment. And I mean, they would apply to opioid and non-opioid treatment options, I would think.
Okay. Yeah, those are very valid points. It's a bit static when you're just reading out things in a chat box.
Do we have anyone who answered the poll who would like to unmute themselves and maybe provide some thoughts on why they answered yes or no?
It's that shyness factor that we encountered this morning.
Mm-hmm.
I won't be shy. I can be not shy. My name's Melissa Pym, and I didn't answer the short answer question because it wouldn't let me type properly. I'm on my cell phone. But I wanted to note that I have a lot of clients who are really sort of opposed or concerned about shifting from their opioid use to a methadone or Suboxone program because of the idea that this will then just be like a new substance use challenge for them. So I think that that's one of the main practical pieces that sort of comes to mind when I decided to join this seminar.
Yeah. Well, thank you very much for that. When we come to the work we've been doing to try to find options here, that was very much one of our concerns. We're looking for something that is a non-opioid and yet has efficacy, perhaps beyond even facilitating withdrawal. And so I'll describe some of the work that we've done on that. At least it's encouraging, but I certainly appreciate the point you're making. Thank you.
And one final answer from the short answer was, I guess it's a question, how effective would it be for emotional pain, which is just as real and as important as physical pain?
Absolutely. Again, you know, that's why we need to do clinical research. Those sorts of questions have to be asked, of course, and then in a sensitive way so that we can get real answers to whether or not it's effective in dealing with the psychological traumas that exist. So that's a really good point too. Thank you.
Mm-hmm. Yeah, and as preclinical scientists, we somewhat naively put these questions forward, but we want to make sure that our research is aligned with the needs and wishes of the community, including.
Yeah. And Lily, that underscores the point I was trying to make in that early slide about the reverse translation. I was emphasizing, you know, getting ideas from the clinicians observing their clients, but it's equally important, if not more important, to get the perspective from the people who are choosing to use these compounds. So, yeah, it's very important to us.
Mm-hmm. Okay, I think maybe we can move on to the second part of the presentation. I'll keep monitoring the question and answer.
Great. Yeah, we have some more. Just
Mm-hmm.
go ahead and get rid of these are the questions. And now I'm up into the second part of the talk, which is on our own work, trying to develop a novel non-opioid pharmacotherapy. And I got into this sort of by following my own curiosity. I'd heard about a development in Vietnam. Michael Krauss had been involved in analyzing this early on, where they had developed a novel botanical formulation, and which became approved by the Vietnamese health authorities for use in facilitating withdrawal from opioid dependence. In Vietnam, it's called Hien Tos 4, but in Canada, as you'll hear in a moment, we've now got Health Canada approval to import this formulation, and we're calling it Tgir, which stands for Traditional Gastrointestinal Relief, but it's the same formulation. And we've been working with it preclinically for many years, and I became much more interested, frankly, in pursuing this when I discovered it had really profound effects on the brain dopamine system. And I'll just describe that to you. So here the goal is to review this preclinical data that we've developed, which has convinced us that it's worthwhile to pursue this in a clinical setting, because we believe it is a novel non-opioid pharmacotherapy for opioid use disorders. And we were fortunate to have a peer review of all the work that we're doing. We've produced 2 papers on this so far, but by far the most comprehensive one was accepted for publication and published at the end of 2020. And if you're interested in the reference, you can read the paper for yourself, but I'm about to give you a synopsis. So now bear with me here, okay? So these slides, the next 3 slides are very similar. And I want to make 2 points here. So we're going to look at the cartoon at the top for a moment. And our work is done with rodents, and unfortunately for rodents, they respond to opioids the same way that humans do, in that they find it attractive, and if you give them an opportunity to self-administer opioids, they will. In this instance, we're looking at the topic at hand is withdrawal. So we forego the self-administration portion of this and give the animal morphine for 7 days. And then we precipitate the withdrawal. Now here's the analogy to what's happening, sadly, on the downtown east side and other parts of Vancouver. We precipitate withdrawal by giving naloxone, the opioid antagonist. Now, the other thing about this cartoon is that these animals are freely moving. They have a tiny fiber that's in their brain that doesn't cause discomfort, and it allows us to continuously be sampling the cerebrospinal fluid within the brain of that rat. And I can monitor it, and then we take that dialysate and analyze it in HPLC, et cetera. But if you look now down, complete the story here. So on day 7, day 8 has two slides next to it. And so in the one hand, we give the naloxone to precipitate withdrawal. Then 30 minutes later, we give the animal Hiantos, exactly the same formulation that's used in Vietnam, and orally administered, so it's acting via that route. So now we come to the data. So the data is in 2 parts. One is what's happening to the chemistry of the brain while we take morphine-dependent rats, we precipitate withdrawal, and we look at how dopamine changes as we go through that process. So it's not really a control group, but it's the one that's not treated with Hiantos. It's in the blue circles. And I think you can see, so we normalize everything to 100% prior to giving naloxone. Naloxone precipitates withdrawal, and then immediately you start to see a significant suppression in the brain dopamine system. And if you just continue to monitor that for 3 hours, 2.5 hours, then you can see that that level is sustained for at least 2.5 hours or so, and then it starts to move up. The remarkable thing is that in the group that got the oral administration of the Hiantos Tgir formulation in green, you can see that the dopamine system is in the suppression, the hypodopaminergia, which is observed in humans following opioid withdrawal. It is reversed, and then it actually drifts up. This is all dose dependent, but we can elevate the dopamine system simply by giving Hiantos/Tgir following naloxone. On the right-hand side of the C, these are all the different indicators of withdrawal behavior, which are intensified. Grooming, wet dog shape, abdominal stretching, rearing, all these are standardized features in a rat, at least, of opioid withdrawal. And you can see in the blue, those are untreated. The ones that are treated with Hiantos, there's an immediate suppression of those withdrawal signs. So in this one slide captures my enthusiasm about this strategy, because we are able to see a change in the brain dopamine system during withdrawal precipitated by naloxone, and we're able to see our candidate botanical formulation reversing that suppression of dopamine and diminishing the intensity of the withdrawal. But we didn't stop there. The next thing we wanted to do is, when we look through the formulation, so there's 12 herbs that are contributing botanical material molecules to this formulation. There's actually 194 different phytochemicals in this mix. We wanted to see if we could find single molecules that we felt would come out of the extraction process and that might be related to work that we were aware of in China on these drugs called tetrahydroprotoberberines. So that little cartoon under B there is one of these molecules called L-tetrapalmitine. And then what we did is we collected plasma from the rat, and we collected CSF from its ventricles, and we subjected, this is after the animal had been given Hiantos, So we're giving the animal the whole total botanical formulation, and then we're looking for a needle in a haystack. That molecule up there, L-THP, is it in the blood and the brain of a rat after it consumes the Antos? And so in C, those are the signature mass spectrograms of that molecule. And then when we look in plasma, we see a very clear indication of the L-THPB. And then on the right-hand side of that graph, you can see a standard where we know that's known L-THP. And then on the left-hand side at the same retention time, you see exactly the same peak, both in blood plasma and, more importantly for us, frankly, in the CSF, the cerebrospinal fluid of the brain. So we were able to find then one molecule out of perhaps 194, but one that has a history of being a very profound analgesic and also is being explored in China as a potential molecule for dealing with substance misuse. So then we did the final experiment we're showing here, where instead of giving the animal the Hiantos, we wanted to see what would happen when we gave L-THP under our circumstances. So again, it's the same protocol as before, 7 days of morphine, naloxone precipitated withdrawal on day 8. You can see again in blue, this is a brand new experiment. It's got replication. So we're able to see our hypodopaminergia induced by naloxone in rats that are dependent on opioids. And then the important one here is the red square. This is the ones that are given L-THP prior to after naloxone, 30 minutes after. And you can see exactly the way Hiantos did. It reverses the suppression of dopamine. And then if you quickly look over onto C, we're looking at those withdrawal signs, red, they're all suppressed. So this one molecule is now replicating what we saw in the entire Hiantos formulation. The blue triangle, this is a known drug which blocks a particular dopamine receptor called the dopamine autoreceptor. And we believe that the dopamine autoreceptor plays a key role in hypodopaminergia that happens after naloxone precipitated withdrawal or voluntary withdrawal. And that when you have a known dopamine autoreceptor antagonist, it acts just like Hiantos and it acts just like L-THP, only its sole job is to block the autoreceptor, those doses. So that's the sort of, to me, this provides a biological underpinning and foundation for the much more important clinical work that's being done in conjunction with this. This gives you some confidence that whatever is in this mixture that 10,000 people have taken in Vietnam as they attempt to manage their withdrawal from opioid dependency, something in there, most likely a tetrahydroprotoberberine, is producing these changes and reversing the brain hypodopaminergia and reducing these withdrawal signs. So what we're going to do next is try to, we're well embarked on this process. We would like to bring this formulation to Canada and also conduct our own clinical trials to confirm its safety and its efficacy. So, but the work to date, again, I'm repeating myself slightly for emphasis, is it's really important that we have a biological signature. So there's very few botanical formulations where you can actually do something called pharmacokinetics to track how quickly an active component is acting and how long it acts for. It's very important in determining, the properties of drugs. Here we have something, one of the components, L-THP. We can now see it in blood, and so we can use that as a biomarker, a biological signature. And I just touched on this lightly because it could be another whole lecture. We believe that the critical part of the dopamine system is its autoreceptor regulation, where it self-regulates its activity, and that gets disturbed during opioid withdrawal. and you get an increased suppression of dopamine, which may be contributing to some of the depressive symptoms that are observed during withdrawal. And dopamine systems are actually very important in determining higher brain function reactions in pain perception. So there's a lot of real importance attached to these observations. I mentioned before that this ingredient LTHP is used in China to provide analgesia and sedative effects. And what we're emphasizing is that it may have an important role in the management of withdrawal from opioid dependence. Now, again, I've been hinting at this. We're delighted. We had a very successful collaboration with colleagues at BCIT that were specialists in phytochemistry, and they prepared a preparation for Health Canada, the Natural Products Division, laying out all of the formulation for what we call TGIR, and it's now being approved for importation into Canada, and it can be used safely in human individuals. But we want to do our own safety trial first to make double sure that this is not dangerous. And secondly, we want to have another look at the clinical efficacy. So we hope to start these activities in the fall of 2021, just as we emerge from COVID, thank goodness. I think we have a pause here, don't we, Lynn? No? Oh, yeah, sorry. So the clinical trial is in collaboration with Michael Kraus and Puya Azar and another gentleman who's a pain specialist, Dr. Kramer. And we'll be evaluating also LTHP itself in addition to looking at Hiantos in our future studies. Oh, this is another, sorry. Before we get to the questions, we also prepared this slide, which Puya had used earlier in the year to summarize some of the relevance of the TGIR approach. And we've already hinted at this, like that this may well be a monotherapy. You know, we heard from one of the discussants a moment ago about people who are reluctant to move from an opioid of choice onto a more legally, really acceptable opioid, whether it's methadone or buprenorphine or Suboxone. And what we're emphasizing here is that we believe that we have a non-opioid alternative. So you're not switching over to something that's simply a substitution of a different form of opioids. And so what this it may be effective in people with a history of opioid use disorder as a non-opioid supplement. And again, I really want to emphasize that we're not saying that this cures everything. What we're saying is that all the evidence in Vietnam suggests that individuals who take this orally as they're going through their journey to try to detoxify and withdraw from their dependence, all the indications are that it helps. And I've hinted at the fact that it may be the properties to regulate the dopamine system that might be contributing to this. But it's not the cure. It's a supplement that can assist. And then the other thing, I didn't emphasize this, but we have some other data that suggests if you give Hiantos in conjunction with morphine, and we've only done morphine as the opioid, it actually suppresses the activation of the dopamine system produced by opioids. So even though there's not an opioid in here, there's no opioid antagonist action, it has the capacity in preclinical studies to suppress opioid-induced activation of the dopamine system, which may be another benefit. So it could be used in people who are experiencing problems when they're transitioning to, or even trying to wean themselves from opioid agonist therapy. It might help under those circumstances by blunting the effect of the opioid. That's a hypothesis. We don't have evidence for this. The other really exciting thing, given our discovery that LTHP is contained within this formulation, we're very interested, and we have active research underway looking at whether or not the hypersensitivity to pain that occurs during withdrawal might also be treated directly with this formulation. And that would be very exciting. And even people that are not experiencing pain because of a dependence on opioids and withdrawal, we want to explore whether this may have efficacy for them too. So this may be a non-opioid supplement. It may be effective in multiple analgesic regimens. It could be used in hospital. People are sustained on opioids after major surgery. Perhaps it might affect in the tapering. And then again, after a lot of work, we hold out the hope that it might be able to be used in outpatient situations. So that's the end of my formal presentation. Just check my clock here, 2:45. So we've got 15 minutes to ask further questions. And there's a point I want to make in a moment, but I want to hear from you first. So Lily, over to you.
Yeah, thank you, Tony, for the thought-provoking presentation. Now we can open the floor to some questions. If anyone wants to unmute themselves and ask a question, go ahead. I didn't see any new questions coming in on the WOVA platform, and we took up all of our poll answers.
So Melissa is here again. I'd love to hear from her.
Hi there. I am actually just curious. I don't think I heard you cover it. If there are any known side effects or long-term effects, and also how long the individual might be expected to be using this therapy into their journey of recovery. And I also wanted to note, just of course, there would be additional supports in the form of therapies and such to address if the substance use was being initiated by the consumer as a strategy to manage, let's say, a trauma or something like that, that obviously that would continue to be part of this.
Those are all extremely important points, Melissa. Thank you. So let's deal with the simple one about the just in the withdrawal context right now, and that's the claim right now, is that this will facilitate withdrawal from throughout as you go through the detoxification process. It's the longest is 7 days. It can be between 5 and 7 days, depending on how well the person responds to the oral therapy. They're treated twice a day. They have more tablets on the first 2 days, and then it tapers down to a couple of tablets twice a day, say for the 7-day period. And that's the end of it. The Vietnamese have done some open trial, like not rigorously clinically valid studies, but they got an impression that if the person is maintained on a weekly regimen, incidentally, their relapse rate after 6 months in the standard detoxification plus their drug Hiantos is well over 60%. And in the individuals that were given supplements of Hiantos over those 6 months, it goes up to about 80%. So it's not, the individuals are indeed relapsing for various reasons, but that's a fairly high success rate. Somebody earlier today, maybe it was Michael, was saying that on opioid agonist therapy, the retention even is quite often less than 50% in buprenorphine treatment and sometimes methadone. So anyway, and of course, that's what we would plan to do, of course, in our test is to do a treatment as usual control to see that it's the TGIR is at least as good as that and hopefully better, okay? But that work remains to be done. We're relying on very good evidence from 10,000 people in Vietnam to date, but Michael and I would like to see this for ourselves before we would recommend that this be used widely. So the other point sorry, I missed you had another point at the end there.
I think I was just talking a little bit in and around like additional therapies alongside of the medication.
No, this is not, just here, take this drug and go away. The whole idea here is that if getting through withdrawal successfully, if you can't do that, it's a barrier. if you can do it, it's a gateway. Well, it's a gateway to all the other things you need. You know, there's a great deal of social support and counseling support and everything that will actually be the real reason why you get somebody through to a successful conclusion on this journey. And we recognize that, you know, obviously. But my point is, and I'm going to raise this in a moment after I hear from others of you, my point is that there's so much difficulty in making that transition that if we can do something that removes that barrier, then we might have a different option for dealing with this problem. That's the philosophy here.
And I'm understanding that we're going to get some feel-good hormones firing for that individual, which would probably be happening much later down the road, even if they did successfully experience like the process of withdrawal.
Again, you're doing a good job here and touching on all the right points. There's nothing in this botanical formulation that actually has its own intrinsic hedonic effect. Okay, nobody in Vietnam is switching their habit from opioid dependence to Hiantos dependence. It doesn't seem to do. And as I said in passing a moment ago, when we're looking at the effects on the dopamine system in the rat, when we give them morphine, morphine activates the dopamine system. And that's probably why it has these hedonic effects. This formulation blunts that. So it's actually reducing the effects of the opioid on these brain emotion systems and brain pain control systems as well. Anyway, thank you for your questions. I hope that helped. So what I may do here, so I'm trying to be provocative in the last few minutes I've got here. Okay.
I think before we move on, can we have Brian? I think he wanted to ask a question.
Yeah, sorry, Brian.
Hey, I actually just wanted to ask questions about the clinical trial process.
Yeah.
Because I know it's ridiculously long and extremely expensive. Mm-hmm. And so based off the trials you talked about in Vietnam, that's around 10,000 people is a pretty big sample size.
Yep.
So then I was going to ask why you would redo a clinical trial, but to me it seems that you want to see if it's like you want to see the efficacy in Canada.
Yeah.
And so did the clinical trials in Vietnam help speed up the clinical trial application process at all, or was it just kind of the exact same process, but you used?
What they did is they had the Vietnamese health authorities wanted them to do a head-on trial against the treatment as usual, which in Vietnam was actually chlorpromazine, a very old-fashioned antipsychotic drug. And obviously it proved to be equal to that, and if not better. So it was a formal clinical trial. The 10,000 people that have been treated subsequently, there hasn't really been any in-depth clinical analysis of that population, sadly. What we know is whether or not they come into the clinic and they successfully transition from opioid dependence to being detoxified, and that they don't have the resources to do the proper follow-on. Now, the reason why, so I would strongly defend our decision to do a safety trial here, okay? Because it's a different population, a different background. The last thing I want to do is to introduce something, raise people's expectations. If there's some, and I'm not expecting any side effect profile, but I want to see for myself that this is safe. And it's relatively inexpensive to do that, maybe a couple of $100,000. And so to know that you're dealing with something and making it safe, we don't have to do that. We have Health Canada approval right now to import TGIR and to give it to people. But as responsible scientists, we would like to make sure it's safe. The second point you raised, which is a really good one, about the inordinate amount of money it takes to do clinical confirmation. Why would you do that if you've already got something that may be working? Our trial will be on a different population. We will start initially with the population of individuals who have become dependent because of maybe it's misprescription, and then eventually they began to misuse prescription opioids, but that's the population that we would like to look at specifically to see whether or not, and that's not one that's happening in Vietnam. It's more a dependence on illicit opioids that then the individual wants to transition from. In our, especially in the United States, many people transition into a dependence on opioids because of their pain management experience with opioids. So we want to look explicitly at that. But now let me address quickly your point about the cost. Because we have the proof already, and we know we don't have to go through all the toxicology trials. We'll do our safety study. We'll show to our satisfaction that this is safe. And then we can begin to look at a population, as I say, that wants to withdraw from their dependence on prescription opioids. And so we're skipping about 3 years out of the 7 years of the clinical trial, okay? And we also have the drug is available to us. If this ever comes onto the market, it will be a modestly priced option for people. And so we're shortening that. But the reason we're doing it is we would like to have gold standard evidence that we can present to people who are living under very difficult circumstances and offer them some real hope rather than something that may be only partially effective. So that's our motivation here. So, Madam Moderator, can I bring out my provocative point?
Go ahead.
So it's the following, and it relates to the whole purpose of doing preclinical studies and doing the translational research that I've been talking about here. So the analogy between what we're dealing with now in the opioid crisis and the AIDS crisis of the 80s has been made several times. And of course, there's a profound link here because one of the vectors to becoming infected with HIV/AIDS was not using clean needles and using, encountering the virus through that vector. And then what happened in order to alleviate the situation is that the public health interventions and the harm reduction approach came in, which is absolutely essential. That first line of attack was able to, exactly in the same way that COVID has been dealt with right now, the first line of attack was the public health professionals teaching people about the presence of this virus and how to prevent contact with it. My point is that, and that was a major accomplishment, but the thing that has revolutionized the treatment of HIV/AIDS was the development of the retroviral drugs. The cocktail, Vancouver was at the forefront of developing that option. And that collection of retrovirals was based on an understanding of how viruses replicate and how you can intervene with specialized drugs to prevent that process. And then you take that, and it turned out that you needed to have a combination of retrovirals in order to really effectively lower the HIV/AIDS titer in the people that had contacted it. And so it's a 1-2 punch. The real impact and the reason why the world is not living with the challenge, it's still a factor, obviously, but the reason why it's not a worldwide pandemic anymore is because basic clinical science was translated into effective medicine and put into the clinical practice and made available at reasonable cost to the individuals so that they could lower their HIV/AIDS contact. So I see the sort of these huge scourges that we're dealing with now. Now you move ahead to COVID, okay? Exactly the same, much shorter time scale. The first line of attack was the public health. They saved us from the initial wave. But then again, the second wave appears, and there may be a third wave. And so what is it that's saving us from COVID? It's the basic clinical science that discovered the improved standardized vaccines, but the real hero of the day here is the development of mRNA vaccines, all done preclinically. nobody wanted to have anything to do with that idea, 10 years ago. And some brave scientists stayed the course, figured out how to do it. They even developed it, thanks to Vancouver-based science, the delivery system for taking the mRNA into the individuals. I've had 2 shots of it, no reaction. The best thing I ever did. So again, the thing that's going to save us from the scourge of COVID is this application of science. So now we come to the third crisis that we're facing, which is the opioid crisis. And what we're dealing with right now, frankly, and it's very important and very welcome, is just the first stage. We're standing in here, and it's a public health harm reduction approach that's having a big impact. It's taking a strategy of substituting safe opioids for unsafe opioids as a means of preventing toxicity and death. But very few people are bringing the same urgency to the advancement of preclinical science into blockbuster drugs that are actually going to be, hopefully, the second punch that's going to help us to really deal with the opioid crisis. So I believe strongly, personally, that we have to have this 1-2 punch. And the other thing that I spend a lot of time thinking about is what is happening to all these individuals who are being resuscitated with naloxone and naltrexone. No one seems to be paying any proper attention to what I referred to briefly earlier about the fact that as you undergo repeated naloxone-induced withdrawal, you're actually sensitizing the pain systems in the body, okay? So these people are not only being resuscitated, it may be aggravating the extent of their biological condition, the biological aspects. Obviously, it's fantastic that we can provide immediate resuscitation, but what happens to the person once they have been resuscitated? And then what happens when they encounter repeated resuscitation? I think we have to be paying much more attention to that. And I think it really underscores the need for providing all the proper support that's needed when someone is so unfortunate to encounter an overdose and then having to deal with it more than once. So those are some of the provocative questions that are rolling around in my head. And I wanted to end on that note, Lily, just to see if it gets any reactions or whatever. Melissa.
Thank you. Do we have any last questions before we wrap up?
We have a hand up. Melissa again has.
It wasn't a hand, it was just a clapping one. It was a hand, but it was the clapping emotion, not the hand. I don't have anything to say. Those same thoughts run through my mind, and I have clients who are using multiple doses of naloxone in a week. So thank you for bringing that to the forefront. I will be doing some independent research there.
Good. Well, I'm going to try to, I'm not a clinician scientist, I'm a preclinical scientist, but I really would like to work on that question because I think it's profoundly important.
Thank you, Tony.
Done. Look at that. Right on the dot.
Time, yeah.
Thanks, everyone.
Yeah, thank you, Tony. Thanks, everyone, for the participation. The recording and workshop documents like slides and the paper Tony discussed will be uploaded online and shared with you. And thank you again for attending, and have a nice day.
Thank you, Lily. Great job. Okay, we'll talk later. Thanks, everyone.